come to biology, the scientific study of life. Predation. Predation may be an important cause of density-dependent mortality for some prey populations. If a predator encounters and captures more food as the population density of the prey increases. As a prey population builds up, a predator may feed preferentially on that species, consuming a higher percentage of individuals. For example, a trout may concentrate for a few days on a particular species of insect that is emerging from its aquatic larval stage. Then switch prey as another insect species becomes more abundant. Toxic wastes. The accumulation of toxic wastes can contribute to density dependent regulation of population size in laboratory cultures of small microorganisms. For example, metabolic byproducts accumulate as the populations grow, poisoning the organisms within this limited artificial environment. For example, ethanol accumulates as a byproduct of yeast fermentation. The alcohol content of wine is usually less than 30% because that is the maximum concentration of ethanol that most wine producing yeast cells can tolerate. Intrinsic factors. For some animal species, intrinsic physiological factors rather than the intrinsic environmental factors appear to regulate population size. White-footed mice in a small field enclosure will multiply from a few to a colony of 30 to 40 individuals, but eventually reproduction will decline until the population ceases to grow. This drop in reproduction is associated with aggressive interactions that increase the population density, and it occurs even when food and shelter are provided in abundance. Although the exact mechanisms by which aggressive behavior affects reproductive rate are not yet understood, researchers have learned that high population densities in mice induce stress syndrome in which hormonal changes can delay sexual maturation, cause reproductive organs to shrink, and depress the immune system. In this case, high densities cause an increase in mortality and a decrease in birth rates. Similar effects of crowding occur in some other wild rodent populations. These various examples of population regulation by negative feedback show how increased densities cause population growth rates in decline by affecting reproduction, growth, and survivorship. This helps answer our first question about populations. What causes a population to stop increasing? Now, let's turn to our second question. Why do some populations fluctuate dramatically over time while others remain more stable? Population dynamics. While some populations appear to be more stable in size than others over the long term, all populations for which we have data show some fluctuation in numbers, although ecologists can determine an average population size for many species. The average is often of less interest than the variation in numbers from year to year or place to place. The study of population dynamics focuses on the complex interactions between biotic and abiotic factors that cause variation in population size, stability, and fluctuation. Fluctuations or populations of large mammals such as deer and moose were once thought to remain relatively stable over time, but some long-term studies have challenged that hypothesis. The moose population on Isle Royale, an island in Lake Superior, is a striking example. Moose from the mainland colonized the island around 19 by walking across the frozen lake. However, the lake has not frozen over in recent years, so the moose population has been isolated from immigration and migration. Nevertheless, as shown in this video, shows the population has been anything but stable, with two major increases and collapses over the last 40 years. In general, the severity of winter loss in large grazers and browsers inhabiting temperate and polar regions is proportional to the harshness of the winter. Colder temperature increase energy requirements and therefore the need for food, while deeper snow makes it harder to find food. Although large mammal populations are more dynamic than once thought, they show much more stability than other populations. The Dungeness crab, as shown this figure, is a classic example of an erratically fluctuating population, as shown here. The crab population at Fort Bragg on the Northern California coast varied between 10,000s and hundreds of thousands over a 40-year period. By contrast, over this time period, the most population of Isle Royal varied only between 500 and 2,500. One key factor involved in the large fluctuations in the Dungeness crab population is cannibalism. Females release up to 2 million eggs each fall, and large numbers of juvenile crabs are eaten by older juveniles and by adult crabs. In addition, successful settlement of larval crabs occurs only in shallow water and depends on ocean currents and water temperature. If winds and currents move larval crabs too far offshore, they cannot reach the ocean bottom and settle successfully. Small changes in environmental variables seem to be magnified by density dependent cannibalism, and together these factors explain the market fluctuations in Dungeness crab populations. These results, as shown this figure, these results support the hypothesis that the dynamics of many populations result from a complex interaction of biotic and abiotic factors. Notice that metapopulation and immigration. To this point in our discussion of population dynamics, we have focused mainly on the contribution of births and deaths. However, as we have read, immigration and migration can also influence population. This is particularly true when a group of populations is linked, forming a metapopulation. For example, immigration and migration link the building's ground squirrel population, forming a metapopulation. How might migration influence population dynamics? A metapopulation of song sparrows on Mandarin Island and a cluster of small islands off the coast of British Columbia provides an example. On relatively island Mandarin Island, differences between births and deaths are the principal influences maintaining the highly dynamic population of song sparrows. In 1988, 1991, only 11% of the breeding song sparrows were immigrants. By contrast, during the same period, immigrants made up 57% of the breeding song sparrow population on a cluster of small island near Vancouver Island. This higher level of song sparrow immigration combined with a higher survival rate on small islands resulting in greater stability in song sparrow populations on the small islands than on Mandarin Island. The metapopulation concept underscores the significance of immigration and migration in the contrasting song-sparrow populations, and is important for understanding populations in patchy habitats. Population cycles. While many populations fluctuate to under predictable intervals, overs undergo regular boom and bust cycles, fluctuating in density with a remarkable regularity. For example, some small herbivorous mammals, such as voles and lemmings, tend to have 3 to 4 year cycles, and some bears, such as ruffed grouse and ptarmigans, have 9 to 11 year cycles. One striking example of population cycles is the 10-year cycles of snowshoe hares and lynx for northern forests, as shown in this figure. Of Canada and Alaska, lynx are specialized predators of snowshoe hares, so it is not surprising that lynx numbers rise and fall with the numbers of hares. But why do hare numbers rise and fall in 10-year cycles? Three main hypotheses have been proposed. First, the cycles may be caused by food shortage during winter. Hares eat the terminal twigs of small shrubs such as willow and birch in winter, and may suffer from malnutrition due to overgrazing. Second, the cycles may be due to predator-prey interactions. Many predators other than lynx eat hares, and they may overexploit their prey. Third, the cycles may be affected by a combination of food resource limitation and excessive predation. If hare cycles are due to a winter food shortage, then they should stop if extra food is added to a fired population. Researchers have conducted such experiments in the Yukon for 20 years over two hare cycles and report two results. First, hare populations in the areas with extra food increased about threefold in density. The carrying capacity of a habitat for hares can clearly be increased by adding food. Second, the hares with extra food continue to cycle in the same way as the unfed control populations. In particular, cyclic collapses occurred in both experimental and control areas, and the decline in numbers could not be stopped by adding food. Those food supplies alone are not the cause of the hare cycle shown in this figure, so we can discard the first hypothesis. By putting radio collars on hares' field, ecologists can find individual hares as soon as they die and determine the immediate cause of death. Almost 90% of the hares that died were killed by predators. None appear to have died of starvation. These data support either the second or third hypothesis. Ecologists tested these hypotheses by excluding predators from one area with electric fences and by both excluding predators and adding food to another area. The results supported the hypothesis that the hare cycle is driven largely by excessive predation, but that food availability also plays an important role, particularly in the winter. Perhaps better fed hares are more likely to escape from predators. Many different predators contribute to these losses. The cycle is not simply a hare lynx cycle. For the lynx, great horned owls, weasels, and other predators, that depends heavily on a single prey species. The availability of prey is the major factor influencing their population changes. When prey become scarce, predators often turn on the another. Coyotes kill foxes and lynx and great horned. The collapse of the predator population. Long-term experimental studies will help to unravel the complex causes of such population cycles.